Hello everyone and welcome to chapter 1.3 cell structure and function. Now, we have already covered 9 different cell structures in the previous class. So let's go ahead and cover the remaining 7. But before we start, I just want to remind you that we need to do 3 things. Number 1, name it. Number 2, recognize it. Number 3, we should know its function. This is the main point of this particular chapter. Okay, the first cell structure we'll be talking about today is the cell wall. And we all know the cell wall is only in plants. It's only available in plants. It's a thick, rigid layer on the outside that is made of cellulose. Cellulose is actually a type of carbohydrate. And we'll talk about carbohydrates in more detail in chapter 2. Now, this cellulose layer is actually permeable, fully permeable, because there are spaces and gaps between fibers. Think of it like a very rigid net. So it is rigid, it has structure, but there's a lot of holes. So things can pass through very easily. Now, think of this in contrast with the cell surface membrane, which is down here. And the cell surface membrane, just a recap for you, is made of phospholipid bilayer. And this phospholipid bilayer is partially permeable. So, plant cells have a cell wall, but it's fully permeable. And the cell surface membrane, which is available in all cells, is what controls the movement of substances in and out of the cell because it is partially permeable. So let's talk about the function of cell wall. How do we put it? Now, it provides structural support. It is like a very rigid layer, right? And it prevents bursting. And of course, because it prevents bursting, it limits the cell size. Okay, I think that is quite straightforward. Let's move on to the next one, which is plasmodesmata. Plasmodesmata is again a feature that is unique to plant cells. They are strands of cytoplasm passing through channels at the plasmodesmata, and at these regions there are actually no cell wall. Okay, very little or no cell wall, and the cytoplasms are connected, so substances can pass in and out freely from cell to cell. Now, these substances include water, sucrose, amino acids, mineral ions, ATP, all those things that are in the cytoplasm can pass through via this plasmodesmata. Bear in mind, this plasmodesmata is kind of small, so don't expect mitochondria or chloroplasts or other organelles to be able to pass through it. Just those solutes in water, just these items. Now, why do we need this? Now, of course, this is common sense. It allows more rapid transport of substances from cell to cell in the plant. No need to have cell wall there. Shorter distance to travel. Anyway, so next one here is about vacuoles. Now, we talked a little bit about vacuoles when we talked about Golgi and how it also facilitates the formation of small vacuoles. However, the vacuoles in animal cells are small and temporary. Vacuoles in plant cells are large, permanent, and central. Okay, and it's not like a vesicle. Okay, it's quite large, it's quite permanent, and the membrane has a special name here. It is called a tonoplast. It is also partially permeable, and it's also made, the membrane is also made of phospholipid bilayer. All the membranes in the cell, all made of phospholipid bilayer, okay? Now, what is its function? This is storage. So this is not very different from SPM and IG, storage of cell sap, which contains water and all these other solutes, ions, minerals, salts, pigments, sugars, etc. It also stores waste products for the cells, keeps it separate from the rest of the organelles, keeps it from interfering with other reactions maybe. And it has this extra interesting function. It pushes the chloroplast to the edge of the cell. Now, why is that? The vacuole contains a lot of water, very high amount of water compared to everything else. And when the when it's filled with water, it gets bigger, and therefore it takes up more space and pushes the chloroplast to the edge of cells, so that chloroplasts are able to is in a better position to capture sunlight at the leaves. Yeah, this also gives turgidity to the cell, again because it has a lot of water in the vacuole. Now these are some more images of the vacuoles and tonoplasts. Okay, again, this is vacuole, the membrane surrounding it is tonoplast. And I just want to point out to you another word here. The nucleus and cytoplasm together, including all these other words, is called the protoplasm. And you will see this term sometimes, but a little bit more rare. I don't know why, but people tend to confuse protoplasm with vacuole and tonoplast, but it's not. Protoplasm is actually everything in the cell, right? Including the nucleus and cytoplasm, and even other organelles. Okay, structure number 13. Centrals and centrosomes. Now, what about centrals and centrosomes? Now, you might have heard of it before, and this would be in the context of cell division. If you haven't, don't worry, I'll work through it. Now, centrals are cylindrical. This is a central right here. You can see we usually actually just draw it like this, but if you look at the structure, it's actually really pretty. Okay, and it's made of all these little tubes called microtubules. Microtubules as in small tubes, and not just one of them. You can see they're in groups of 3, and then there are 9 groups of them if you count. So 1, 2, 3, 4, 5, 6, 7, 8, 9. Yep, 9 groups of 3 microtubules, and this makes up a little cylinder. Okay, and by the way, it's not found in plant cells. Now, you might have heard the term centrosomes also. Okay, this is a pair of centrals. They form a centrosome. So be careful what terminology you're using. Anyway, let's talk about the functions of centrals first. Number 1, it is involved in cell division because it replicates from this, okay, from one cylinder to form 2 cylinders that are right angled to each other. This is how we usually draw it. 90 degrees here, you can see. And this replicates before each cell division and moves to opposite poles of a cell. Notice the language here. We always talk about the cells in terms of poles, not ends, not sides. Opposite poles. That is a proper term. Now, what else? Centrals are also found elsewhere in the cell, especially at the flagella and cilia. These are modified a little bit centrals, and what they are in charge of is to act as a microtubule organizing center. Yes, centrals are made of microtubules, but it also functions to organize other microtubules. So this is called an mTOG. And again, like the name shows, it organizes and assembles microtubules. Now, how about centrosomes? How do we distinguish it from centrals? Again, centrals is just one cylinder, right? Centrosomes are 2 cylinders right angled to each other, which form during cell division. And you can see them on this side here and this side. You can see it here and here. We will go through the stages of mitosis later on in other chapters. Don't worry about it first. But its function of centrosomes is really to function as an mTOG as well. Why is that? Centrosome is also considered as an mTOP specifically for cell division because it also organizes and assembles microtubules for the formation of spindle fibers. Spindle fibers are also made of microtubules. Sometimes they are called spindle microtubules, right? And this is, you can see here that the mTOPs, which are centrosomes, are forming the fibers to connect to the chromosomes here. Again, we will learn more detail about cell division and mitosis in chapters to come, so do not worry about it. But for now, just know that they assemble microtubules during cell division for the formation of spindle fibers. We will talk about the rest in future chapters. Now, let's talk a little bit more about microtubules. These are also considered cell structures. They do not only exist as centrioles and centrosomes or even fixed spindle fibers. In fact, they are found in many places in the cells. Right, so again, what are they? They are small tubes, and when I say small, I mean very, very small. They're like around 25 nanometers in diameter, and they're made from a protein called tubulin. Okay, and these tubulin form dimers, okay, and then they polymerize, so they form chains like this, and then the chains then form sheets, and then these sheets close up to form a tube. I wish there was an animation here, but I couldn't find one. If you find one, let me know. It's not very important to know the terminology here. It's just for your information. But anyways, what you need to know, okay, is number one, they're small tubes. They're made from a protein called tubulin, and they are formed and broken down at microtubule organizing centers. We saw two of them, all right? We saw centrosomes already, which is in charge of forming spindle fibers during cell division. And we also saw how centrioles, other than forming centrosomes, can also form, can also act as mTOPs near the flagella or cilia. We'll see a diagram of it later. Okay, but what is it for? As I said, other than the centrioles and the centrosomes, they are found everywhere. All these, areas labeled in green, these are all microtubules. Can you imagine? All right. They actually make up something we call the cytoskeleton. Cyto means cell, skeleton means skeleton. So the cell skeleton together with other actin filaments. Actin is also another type of protein, which we will learn more about in A2. However, and the point is this, they make up the cytoskeleton, so this provides mechanical support, and other than structure, it also acts as an intracellular transport system. What do we mean by that? Okay, think of train tracks. How do components or vesicles know where to go, right? Which pathway to take there? So cytoskeleton here, made of microtubules, acts as an intracellular, so inside cell, transport system in order to aid movement. Okay, let me just show you a little video on cytoskeleton. Now this here is actually the microtubule. This is part of cytoskeleton, and you can see there's actually many of them around the cell. And you can see this vesicle, which is this blue blob here, being dragged along by this motor protein. This motor protein is not part of a syllabus, but look how cool it is. It's like walking on the cytoskeleton, bringing the vesicle from place to place. Think transport vesicles. Think of secretory vesicles we learned this chapter, right? Like this really shows us how they are transported exactly in the cell. How cool, right? Look how cute it is. It's like almost comical seeing it move. Of course, this is a CGI kind of video. It is animation, but it is based on research. Anyways, back to the topic. Again, we are talking about microtubules. They make up the cytoskeleton. Other than providing structure and support, they are an intracellular transport system. Now, they also help in movement of the cell. So as the cytoskeleton changes, the cell shape changes, creating movement. So like the beating of the flagella, okay, this is also because of microtubules. And of course, because microtubules make up centrals and centrosomes, these mix up spindle fibers as well, used in cell division. Okay, so we have mentioned this a little bit just now, but for our final two cell structures, we'll be talking about cilia and microvilli. Now, cilia has also something to do with microtubules, right? It's actually a complicated structure right here, and you can see how microtubules also form a cylinder-like structure here, and has, and as we mentioned just now, has an mTOC, a microtubule organizing center at its base, in order to control movement. Now, cilia is not to be confused with microvilli. It is smaller in diameter, and also not to be confused with flagella, which is mostly found in prokaryotes. Flagella, we always think of it as found on bacteria or archaea. Yeah, so not flagella, it's not microvilli, it's cilia. Cilia is motile, cilia makes the cell motile. It moves rhythmically, and its function is really for movement and locomotion. I wrote here, this here is actually a paramecium, which is actually a eukaryotic organism. Yeah, it's a unicellular, so it's just one cell, microorganism, but it is a eukaryote because it has nucleus. Now, you can see how it uses cilia to propel itself forward. How cute it is. It's like little legs, right? Move, move, move. Anyways, it could be found in paramecium, but also it's found on ciliated epithelial in our lungs. Now, in our airways, in our trachea, there are ciliated epithelial cells, okay, which sweep the mucus produced here. You can see this in yellow. Sweep it upwards, and they sweep. Okay, it moves in a rhythmic motion. And this is all because of the microtubules, which are controlling those movements inside. As the microtubules change in structure, therefore the ciliated epithelial cell moves. So yeah, that's cilia. Last cell structure, microvilli. Now, microvilli is found in animal cells only. Okay, so not in all eukaryotes, but only animal cells, and it is bigger in diameter. It's found on the epithelial cells in intestines and kidneys, and they are kind of like finger-like extension of the cell surface membrane. Now, unlike cilia, which is mostly for movement and locomotion, right, microvilli, the structure really aims to increase surface area of the cell membrane. For what? Well, for things like absorption, secretion of enzymes, digestion at cell surface maybe, or even excretion of waste substances. Now, increase in surface area would always mean increase in the rate of diffusion or diffuse? I can't spell. Diffusion or just movement across the membrane, because there is no membrane, so that's easier to move, right? And this would really increase the rate of all these processes as well, the efficiency of these processes. So yep, that is the last cell structure. Now before we end cell structures for, well, eukaryotes, we need to have a little bit more thought on this process called centrifugation. Now centrifugation is the idea of rupturing cells. So we maybe use some soap solution or something similar like that to rupture the outside cell surface membrane. Okay, so we get a cell extract, which is everything inside the cell, and then we spin them at really, really high speed using a machine. So what happens exactly in light of all these cell structures? What would happen is that larger structures will sediment first. What do you mean by sediment? It will sink to the bottom. Okay, let's have an image version of what's going on here, right? Here there is a suspension of broken cells containing subcellular components. This is our cell extract, right, that we have gotten from rupturing cells. We centrifuge it, we spin it at high speed, 800 G, so G is a unit here, so 800 gravitational force, I think, G, for 10 minutes. And we realize what would sediment, this is the sediment. What would sediment is the nuclei first. Why? Because this is the largest organelle. Yep. Okay, then we take this fraction, right? So the liquid that's still up here, that still contains stuff inside, right? This is called the supernatant, fancy term for you. Okay, we take it and we again centrifuge it at an even higher speed for some time. What do you think will sediment next? So if you know your your organelle sizes, sorry, not cell sizes, organelle sizes, you would know that if there's chloroplast, it would sediment first. Okay, chloroplast, mitochondria would be next. Let's just ignore this. This came from a different textbook, right? Just chloroplast and mitochondria will sediment next. Now, other larger organelles may sediment as well, okay, but mostly chloroplast and mitochondria. Now, what if we spin it at even higher speed, spin this fraction only, okay, not the sediment, just a supernatant, for even higher speed for longer. So instead of 10 minutes, we spin it at 60 minutes. Now, this would result in sedimentation of plasma membrane fragments and endoplasmic reticulum sediments, because even though they are only 7 nanometers thick, right, the membranes are all 7 nanometers, but there are a lot of them, so maybe they all clump together and then they will sink. What would sediment next? Last but not least, okay, if you spin it some more for even longer, for even faster, last but not least, you will finally get the ribosome. Why? Because this is the smallest organelle of the cell. So, what is the point here? Do you need to know this? Yes, you do. It came out in past years before. But not so much memorizing each and every sediment, but being aware of the sizes of the organelles. You will see this, you will see an exercise in a worksheet that will help you with the sizes and just being aware of them. So yep, that's centrifugation.